Start recording. We'll have a quiz on Friday. We have lab this week. If you still have to get it done, if you've already gotten all your data, you don't need to come to lab. I have some people coming this afternoon, so if anybody wanted to come and work this afternoon, they could instead of Wednesday. You could all be each other's lab buddies. But I already have enough people for lab. I don't need lab buddies. We've got people we need for that. Let's see. I think that's it. I've got office hours today. From 1:15 to 3:15 p.m. And then let's see. I can have office hours tomorrow by appointment. And that the range that I'm available would be 10 to 2:30 p.m. there. I have somebody coming by at 10:30 already, presumably. So I'll be here then as well. Questions? Okay. All right, then what we've got, so we've got the codon table here, and I wanted to remind you that, so we've got all organisms use, we talked about this, all organisms use the same codon table. Okay, we have some minor weird exceptions, but all organisms use the same codon table, but they do not use the same codons.
Like in terms of frequency?
Use the same codons for a particular amino acid. When there's a choice. So in other words, 4 amino acids that have codons, when you have multiple codons like you do for arginine, right? We've got 6 for arginine. Different organisms use different numbers or amounts. But so that seems a little inefficient that you have all these different codons. But so if you think about it, you know, you would have to make 6 different tRNAs to correspond to our 6 different for arginine. Seems a bit inefficient, but there are ways that nature takes advantage of, we could call it slight mispairing for efficiency. So if we look at a tRNA, here is a 2-dimensional model of it. I'll show you a 3-dimensional model in a second. But what we have here is we have what's called an anticodon loop. And this pairs with the codons on the mRNA sequence. All right, and up here we're going to have an amino acid attached. Let's not say up here, let's say 2,3' end of the sequence. And it's important that that amino acid correspond to whatever the codon is that this matches up with. One thing I want you to see here, and we'll actually probably see it in figures below as well, that if I have an mRNA here, this and everything's, whenever you have pairing, it's always 5' to 3', it's always anti-parallel, right? So if we think about the codon sequence, the codon would go 5' to 3' in this direction. So this would be a G C, and we'll talk about I in a minute. Okay, I'm going to go ahead and call it an A. All right, but it could be anything, or it could be one of three things. But so that would be, in other words, the codon is, you can just flip that, is GCA, which if we look up here would be GCA, it's alanine. Okay? What was that?
Which one is the codon sequence and which one is
The codon sequence is, this is the codon sequence. It's the same sequence. It's just written backwards because it's anti-parallel with the anticodon, the way the anticodon is drawn on this figure. Does that make sense? So they're the same sequence. Yeah.
Can you explain again why it's shaped like that, the tRNA?
Well, it's not really shaped like that.
Oh.
Okay, in 3 dimensions. Okay, but you have this base pairing is going on to cause these pieces to be together. And you've got these arms and they kind of fold over each other. And let's see, I should have a 3-dimensional picture real soon. Right here. Let's bring that up.
Yeah.
All right, so maybe I'll just paste it here. Is that big enough for folks?
Yeah.
I'm not expecting you to get a lot of detail out of this, but it's not these 2-dimensional models you see actually look like this in 3 dimensions, where your amino acid attaches here, and that's your anticodon loop. So let's go ahead and at least annotate that. So this is amino acid attaches here, and really, amino acid attaches here. And this is where our anticodon loop is. Okay, and so we've got, you could think about this in the context of, in our pictures, when we show this in the ribosome, it's going to look like kind of probably a straight up thing, but the ribosome is designed, you've got mRNA, it's going to be feeding through over here, and you're going to be building your peptide over here. It's not really important that you know the shape of that, really for any context, unless you end up being a specialist in the field. Questions? Other questions? Okay. So that's what we've got for this tRNA. And so like I said, and actually as an example here, first off, one thing you can see about these tRNAs, and I think we mentioned in first semester, but maybe not, is that you have a bunch of non-standard bases here. All right, I'm not going to worry about what all the shapes of these things are. We will worry about inosine. We'll look at what inosine looks like. And we will worry about, well, we don't worry about it, but thymine, okay, instead of uracil there. But so these tRNAs will get made, and then certain bases will get modified. And the one we're really going to be concerned here with is inosine, because it can pair with multiple other things. And so what inosine is going to look like, looks like I didn't include it here. Let me give you the base for inosine. Yeah, okay. It feels wrong. Oh, it's missing an N. Okay. It still feels a little wrong because I don't have double bonds in it, but it looks right. Okay, so that's what inosine looks like, right? Which is different from our other bases.
What did you call it? A non-standard?
It's a non-standard base. It's not one of our 4 we think about. But it does get used fairly frequently in terms of tRNAs. We see a bunch of these.
Is it only found in tRNA or just mostly
found in tRNA? It's a breakdown product of, when you think about a purine, when you get purine metabolism, you'll go through inosine if you want to go back and forth between A's and G's. So you see it in metabolism a bunch too. I'm not sure where else you see it. You certainly don't see it in DNA at all. And I frankly don't know a lot about all those, the 100 or so specialized RNAs that people have found in the last 20 years. I don't know how much you would use one of these. It might be useful if you think about like an RNAi or something. You could have more flexibility in recognizing things. That might be useful. It might be toxic. But in terms of the anticodon here, so the anticodon doesn't have to match the codon exactly in terms of the last base. When I say last, I mean the 3', and why did I say nucleotide? Okay, so we don't need to exactly match that. And what this does is this saves the number of tRNAs that are necessary. So that need to be synthesized to cover all the amino acids. And so I don't have that table. You may have noticed when we have the table for the codon usage that things that were right next to each other, they tended to go in pairs. So if we think about the codon, I'm going to scoot back up and I'll be right back down. If we look at this, you notice these two go together, right? Where we don't have a amino acid monopolizing on all 4 of the codons in one of these blocks. You notice the blocks go by the first 2 bases. They go 2 and 2 usually, okay? Or they go 3 and 1, but they're generally, we've got a lot of 2 and 2s, and if you go back and look at the notes for the usage, they tend to, like, you'll have two next to each other, they're pretty similar, okay? And that's because they're probably using the same tRNA, so it doesn't matter which it is.
How did they figure this out? Like, what did they do to research this?
I don't know what the experiments were here. And I think Crick was involved with this, but I don't remember. I mean, yeah, I don't know. I don't know exactly when they figured it out. So I also don't know what technology they had available. But they probably didn't have DNA. Probably did not have DNA sequencing. Yes.
How do you spell the non-standard base segment? Like
Just non-standard.
no, like
Oh, inosine. Like that.
You said a Z, that's an A.
Okay, so we've got so what you've got here though is your anticodons don't have to match exactly, and so what you have is you have some of them where they do actually have to match exactly. So if you do one of these, if you have in that 3rd position of the anticodon a C, it's only going to pair up with G, and if you have an A, it's only going to pair up with U. But if you have a U in that position, you can look at A or G. Okay, and if you have a G, you can look at C or U. Okay, so look at or match up with. And then you can also have cases where you have 3 that are recognized if you put that inosine in. It can actually recognize A, U, or C. All right, so where is that useful? Well, if we go back to the codon table, let's take an inosine. Let's look at how it's arranged. AUC, right? Let's hold on to AUC. And do you see right here? Right here we have this triple and AUC, right? Okay, so we can recognize this with 1 tRNA. So let's say can be recognized by anticodon, what would that anticodon be? Let's figure it out. So our anticodon loop's going to come around like this, right? It's going to be 5' to 3' in this direction. And so our anticodon would go, and it's going to, these are all 5' to 3', right? Like this. So it's going to go I, okay? I, what matches you? So here we have to do an exact match. A, U, because it's RNA. Okay, so it's this position that we call this the wobble base position. Okay, and note, before the I was here, right? But for that other diagram, the 5' and 3' ends have been flipped. Does that make sense? So the I is going to be on the 5' end, whichever way you're drawing it. I'm trying to make my anticodon here match up with my codons the way they're written. Okay, let me ask you a question then. So the anticodon, or there are no tRNAs with an anticodon that goes, I got to get this right, 5', in my head I've got to do 5', I, U, does not exist in nature. Why?
Because it's a stop codon.
Does it match up with a stop codon? So we're looking at IUU. Remember, this anticodon, these are 5' to 3'. I just wrote that 5' to 3'. You have to flip one of them. I recommend flipping that one and then comparing it.
What is I?
I is on inosine and it matches 3 of them, right? It's AUC. You don't need to memorize that.
So is that why it doesn't exist in nature?
What was that?
Is that why it doesn't exist in nature? Because if you had I first, then you would have too much variability in what amino acid you would use. But it's not.
Yes, but be more specific than that. Be really specific with the table. I don't think I messed this up.
You could have a start or a stop.
Okay, let's remember that I wrote this as 5' to 3', right? And things are anti-parallel, so you need to flip it. So it would go, comparing it to these, you'd be going U, U, I. Where does it match up?
So last with A, that's different from when it ends in U or C.
Right, so what we're saying here is it's going to match these 3, right? So I now have a tRNA that would be loaded with an amino acid, but crosses 2 different codons, right? So let's say we loaded that tRNA with an asparagine. It would be putting an asparagine instead of lysine sometimes.
So that's the only instance where that happens, that combination.
Well, we couldn't use an I for this set of codons, right?
Yeah.
We couldn't use an I that corresponded to the CA, or the anticodon for that. We couldn't use one here.
Okay.
We couldn't use one here.
Okay.
We couldn't use one here. So a bunch of places where we can't use it. All right, does that make sense?
A little. If you like type it out, you guys just see this.
I will type it out, but I'm going to have to move it away from the codon table.
Yeah.
Or maybe I'll just have to make room and copy the codon table. But,
So
okay.
kind of like in organic chemistry where you can't use certain things to be like either with them or literature.
All right.
Sure.
So the answer here is Y. And let me, while I type it, why don't I bring down the codon table so you guys can look at that while I'm typing. All right, so it can't be, and that's because that codon would match both, that anticodon would match both the asparagine and lysine codons. Let's see, the anticodon. So both, let's see, it's asparagine, right? And K codons. If it was low, let's say if it was loaded with asparagine, it would sometimes put this in proteins instead of lysine. And what's kind of interesting, well, I'll just be distracting. Something interesting related to histones, but questions? Yeah.
Maybe. So, I guess I'm just like kind of like trying to think like how do 3 of the bases make like an amino acid? Like, how does like AAA make a lysine? It doesn't make it. It just codes.
It codes for it. It's 3 bases. The tRNA is an adapter. So if you think about, if this was our ribosome, okay, if this was our ribosome and here we had A in front of it, let's say GAU, GUC, etc., that the tRNA is matching up. So we're going to go AAA. So we'd have, so this is 5' to 3' in that direction. And so if that's the way I'm going to go with it, then I have to have my tRNA needs to come around. And this is the opposite of the way our figure was, right? So I'm going to try to draw a clo relief, and it's just going to be awful. Like that. And then this is our 3' end, and this is our 5' end up here. This is going to go inosine, uracil, like that. So this is an adapter, and then up here on the 3' end, then we would have, say, a asparagine attached. Let's actually, it's going to be actually attached through, we'll see this actually today if we get to it. So there's my asparagine, there's my side chain for my asparagine. Like that.
Okay.
So this is matching this, and then the protein's being built up here.
But what is that telling?
What is this telling it? This is reading. This is our mRNA, and this is how we're reading the mRNA with the right amino acid.
Oh, right.
And we're gonna talk about the ribosome. We'll have some better cartoons later that may make that more clear. Yes.
You have the other chart where you would kind of like
Where we had these.
no, a little higher. Yeah. Okay. Why did you only box in those 3? Why would the I not also be useful for like?
The I does not pair with cheese.
Okay. Never.
Big no. Never.
Why?
Why? The shape. It's the shape of the mRNA plus whatever the pocket is where that's sticking in complementary with the ribosome. Okay. Other questions? All right, that's a good lead into either what I was going to talk about next or what I'm going to skip to next because, yeah, that's what I'm going to skip to next because it's a good lead in. All right, so way back, I did not talk about the three parts of, let's talk about the three parts of replication that ensure that you have good fidelity. So three mechanisms. Okay, so we're talking about here, how do you make a perfect copy of the DNA essentially? And this is for, and we've talked about this before, but this is bacterial we're talking about here. Okay, so I'm going to give you numbers. I expect for eukaryotes, and probably across eukaryotes, the numbers are different, and I expect for eukaryotes, they're going to be higher because the genomes are larger. All right, so, but the first one here is that the bases pair up, but in the context of the active site of DNA polymerase. A only pairs with T, and G only pairs with C, but in the context of the DNA polymerase active site. All right, so what do I mean by that? Well, you can make these base pairs, A with T and G with C, but you can make a lot of other base pairs, okay? There's all sorts of ways you can arrange these and actually get base pairing. And when you look at the tRNAs, you actually see some of this. All right, so the tRNA doesn't have to be as perfect about getting you the double helix. It also doesn't happen in this same context. So if you look at this, if we think about the shape of the active site for both of these, do you see that if we were to think about what shape these would fit into, do you see these have kind of similar shapes on the outside? We go from this is one backbone here, and this is a backbone here. And if we were to draw those same things over here for this GA pair, right, what would happen? Well, we'd run into the A, right? So the A won't fit. Over here, we've got a bunch of extra space. Maybe that'll work, but you got extra space and you have to polymerize somewhere too. So that's probably offset. And then the same thing over here. Okay, so what this means is only AT and GC fit well in the active site. Does that make sense? So out of this, from this alone, only one error is made every 10,000 or 100,000 base pairs, or bases, nucleotides added, let's say. Okay? Now, this is bacterial, so let's just think about E. coli. I'm not going to italicize it, too much work. But the E. coli, the E. coli genome, anybody know how long it is? I want to say 3 million base pairs. There are multiple E. coli genomes. E. coli doesn't mean bacteria. But
no, I think I was thinking about the different strains.
Okay. I don't think different strains are going to have very different genome lengths.
I just know that the deadly one has 1,000 more of something
Okay.
than the other one.
Okay.
It has 100 what? What's the difference in like the nucleotides? They have like the same amount, just a different.
Well, it could be 1,000 different too. From my perspective, I don't care because it's 3 million, I think. So it's 0.1% different. So might be it's 1,000 more. It might be it's not 1,000 more.
No, I think Maddie's right.
Okay. But the E. coli genome has, I think it's I was going to look it up before class and I forgot to. Has, but let's say 3 million base pairs. All right, so let's take the lower end here. How many errors is that?
4.6 million.
4.6 million? So let's say 5 million. Thank you. Okay. So now I've really swamped out any 1,000 errors because I added, what, 400,000? But so let's say it's about 5 million base pairs. And so from this alone, we have one error. If we have 10,000, what is that going to be? A million is 1,000 times 1,000, right?
Mm-hmm.
So if I add 100, so 100 times 5, is that 500 errors?
Yeah.
I see some confused looks, so why don't we do, let's do actual math to it rather than doing it in our heads. So the question is if you make one error every 10,000, okay, and if we have 5 times 10 to the 6 base pairs, and we make an error every 1 times 10 to the 4th, yeah, I agree with my math before. That's going to be 500 errors. Okay, so you think about that every time the E. coli divides, right, we're going to end up with that many errors. And what happens when you do this? You've got, so is it that I've got my E. coli right here and it's going to divide and make another one. I get 500 errors when a division occurs. Do I end up with errors in just one of these or both of them? Both, because you have one parent strand for each one you have copied, 5 million base pairs. So both end up accumulating 500 errors. So that's a big deal, right? It's not that you make an offspring and it dies sometimes. Sometimes it doesn't matter. It's that both could die off of that. Okay, so we need another mechanism to help fix some of these. All right, so the next thing is DNA polymerase has proofreading activity, this DNA polymerase that they're using. I'll show you the whole figure and then I'll blow it up.
This is the second.
Yes, let's make it 2. All right, so this slows it down. But what it does is it adds a base and then it backs up and it has 2 active sites. The enzyme has 2 active sites. It adds a nucleotide. Then it backs up into the second active, so the DNA is in the nucleotide just added in a second active site that checks the nucleotide. Okay, if the second active site is not a match, it removes it. Or if the added nucleotide is not a match, it is removed. All right, so let's go ahead. That's the process that goes on. Let's go ahead and look at the cartoon for it. Let me scoot down here real quick. Make this big. Okay, so we've got DNA polymerase 1. It has an active site, and then it has a 5' to 3 or 3' to 5' proofreading exonuclease site. What does an exonuclease do? Cut something off the end, so that nucleotide that might not match. So C has a rare form, tautomeric form, right? A tautomer where the double bond moves and you end up with a hydroxide. Remember tautomerization from organic? Okay, so what we've got here is a rare form that will actually pair with A. All right, so this is where we get a lot of our errors from. So this comes in, it will sometimes pair, and then it flips. But it's a rare form, so it's it's flipping back and forth. It's dynamic for the tautomerization. So it pairs because of the rare form, and then it flips back to the regular C. That got incorporated by accident in the active site before the polymerase moves on. What it does is it backs up so that we now can check in this active site. So added, now it's got back up on the DNA. Check this active site. If it doesn't match, what it's going to do is remove that. And then it'll come back and add the right one. That makes sense. Okay? So it's chugging along like this. And it slows it down, but it's really important that you don't have 500 errors for E. coli genome. Yes?
For the first method, how is that like a mechanism to like ensure, like with the first one, like A pairs with T and G pairs with C, but it's not perfect, so you have other things.
So it's A pairs with, the important thing is A pairs with T and G pairs with C, but they have to fit in the active site.
Right.
If you just take A and T and throw them in, A, T, and G and C and throw them in solution, you would just have this happen outside of the enzyme active site, you'd have a lot more errors. So you get one error every 10,000 to one error every 100,000, something in that range, roughly.
And this helps like minimize.
This is going to get rid of, so of all those errors, this, I haven't talked about this, this fixes 99% to 99.9% of the errors.
And that's different than the mismatch system?
That's next.
So yes, so then 99%
Yes.
of that and then 99% of those errors left.
Right. So now we had, what I'm going to do is I'm going to tack these errors onto the end. So our error range is going to get larger, and we're going to focus on the lower end of that. So remember we had 1 in 10,000, and we had 1 in 100,000. Okay? So if we get the best, if we apply the best to the best, right? What we've got is now we've got 99.9% or 1,000 times better. So I'm going to add 3 zeros. You guys agree? Okay. So now we're looking at 1 in 100 million, right? We had 100,000. We added 3 more zeros, 100 million. And over here we had 99, so we made it 100 times better. So I'm going to add 2 zeros. So now we're 1 in a million. Yeah, so we have 1 error. We have 1 error in 10,000. Let's just go with the 99%. 99% of those errors we fix. So now we only have 1 error in. So I took 99%, 1 out of 100, and I added 2 zeros to make 1 in a million. Does that do it, or?
Well, then what's the other part?
The other part is so I've got a range here,
a
10-fold range, and I applied the best part of the range to the best part of the previous range. So I'm getting a bigger, wider range here of where errors might be.
Right.
So I would say that if you only make 1 error in every 100 million for a genome that's about 5 million long, that's probably okay. Depends on how much energy is involved in making sure you haven't made any other errors. Okay, but that's still not good enough, at least on the lower end, right? If you make 1 error in a million, you have 5 million base pairs, how many errors are you going to make? 5, right? Okay, on average 5. Still not good enough. So now we have the DNA mismatch repair system that we learned about earlier. Okay, so the third 3, the mismatch repair system we learned about earlier. This is what the methyl groups with methylated DNA was on your quiz. Well, I'm going to say remember methyl groups. That we learned about earlier. And here we get another 99 to 99.9% improvement. Or let's say, better yet, here 99% to 99.9% of the errors are caught again. All right, so we were at where we had 1 in 1 million was our lower end of our range, right? To 1 in 100 million. That's our range. And now we're going to fix, we'll do the lower end on the lower end again. So now we're talking about 1, I'm going to add 2 zeros for 99%. So now we're at 1 in 100 million at the low end of our range. To 1 in, now I'm going to add 1,000, another set of zeros. What is it, 100 billion? Okay. And this is good enough for a genome of 5 million base pairs. All right. So here, if you're in 100 million, it's like 1 in 20 offspring, or 1 in 20 replications. Did I miss one? So which one are you wondering about, the 10?
The 99.9%.
So I have 3 zeros to this.
Okay.
So it should be 100 billion.
It's 2 of them.
I'm applying this here and this here just to add to the range there. But what?
Well, you had 2 zeros to that one, 3 zeros to that one.
Because I've taken, as we've gone along, I've taken the lower, we've got ranges for all these things, right? And they were all tenfold, were they? Or were they worse? They're better than that. That was tenfold, that was tenfold, and the first one was tenfold as well. And so I was taking tenfold, I was taking the lower end of the range, and each time I was piling in the lower end of the range onto it. So the repair rate, the actual repair rate, based on how somebody's done experiments on this, right? You could do it with DNA sequencing, right? You could grow a bunch of bacteria, you could sequence their DNA, you could see how different they are. All right. And so what they've come up with from that is a tenfold range for each of these steps. And so I took the lowest part of that range and followed it down to here. So I assumed the lowest part of the range was correct for all those. And I got this number. And I assumed the highest part of that range, the highest fidelity, was correct and took it over here.